Okay, so here's the second part of the lab material where we went over the cell cycle and the terms associated with mitosis. So let's just kind of go through the cell cycle quickly here. So everything that you see in blue or greenish color is representing interphase, which is the portion of the cell's life cycle when it is not dividing. Okay, and what it's doing during that time is, for the most part, it's doing whatever its job is in the body. Okay, so if this is a liver cell, it's doing all of its liver cell jobs. If it's a muscle cell, it's doing its muscle cell jobs. Okay, so beyond reproduction, the cell has a role to play in the body, and during interphase is when it's fulfilling that role. Okay, now with respect to further production of cells, there are some important aspects of interphase that we need to talk about, and that's why interphase can be divided into these three subdivisions. Okay, so right after the cell is born, it's a baby cell. It's small because it just got formed by another cell dividing in half. So it doesn't have as many organelles as it might need to really do its job, and it may be kind of inefficient because of that. So what does it do? It starts to grow. It grows, it accumulates more organelles, it makes whatever it needs so it can be fully functional. So that's why the first phase of interphase is referred to as G1. Okay, it's a growth phase where the cell is essentially recovering from the fact that it's just been born. It's growing up so it can do its job as efficiently as it needs to. Okay, now if the cell is signaled that it's going to reproduce at some point in the near future, it will move from G1 into S phase. So this kind of bluer area here is representing S phase. And S phase is the part of the cell's interphase section where it is doing DNA synthesis, and that's actually why it's called S phase, because of synthesis. So we would call DNA synthesis DNA replication today. So we take our original genetic material that we had in the cell and we duplicate it. So by the end of S phase, there are 2 complete sets of the genetic material in the cell so that it's ready to divide. Okay, then if it gets the signal to continue further, it moves into G2. And that's this greener section here. Okay, so G2 is the interphase period where the cell is saying, okay, I'm going to be dividing soon. That means I need to make some proteins or other structures that I need to have inside of me so that when it's time to divide, I'm ready. Okay, so it'll make a whole bunch of microtubules, so it has material to make the spindle. It will start making proteins called histones, so it can wind up its DNA. It'll do all this stuff so it can be ready for the mitotic phase when it occurs. Okay, so this illustration is actually sized the way it is to show that interphase is really most of the life cycle of the cell. Okay, the little skinny yellow part over here is representing mitotic phase. Okay, and mitotic phase is a very short period in the cell's life cycle, and it's the part where the cell is actively dividing. Okay, and it can be divided into mitosis and cytokinesis. Okay, and the two overlap with each other a bit, and we'll talk about that overlap in a moment. But at the end of the mitotic phase, you will have that original cell being divided into 2 new cells. Okay? Now, mitosis is the process where we have division of the nucleus. Cytokinesis is the process where we divide the cytoplasm and its contents. okay? Mitosis is very precise. It happens exactly the same way in every cell, every time, so that each cell ends up with an exact copy of the original cell's genetic material. Cytokinesis is not as exact, and it doesn't need to be. The cytoplasm is roughly divided in half, but one cell may get a few extra ribosomes, the other cell might get a little more rough ER. It's just whatever happens to be on one side of the cell goes into, on one side of the original cell goes into one cell, whatever happens to be on the other side goes into the other cell, and then the growth phase that they go into, the cells go into at the beginning of interphase takes care of anything that they need to replace. Okay, so let's look at a little terminology here. We have a picture of the genetic material here in one form, and then we have a picture of the genetic material in another form over here. Okay, so the illustration that we're looking at here is all of these little squiggly lines are representing the form of the genetic material that you would have in a cell when it's in interphase. And this form of the genetic material is chromatin. Okay, so chromatin is loose and squiggly. It's like a big pile of cooked spaghetti with the strands all kind of mixed in with each other. Okay, so you can imagine that it would be easy to get at the information here because it's all loose and unwound, but it would be really hard to divide it equally if it was in this form. So when it's time for division to occur, the chromatin has to convert into this form, which is chromosomes. Okay, so instead of having a big pile of cooked spaghetti, now we have these little easy to move segments that have this distinctive X shape in the early mitotic cell. Okay, so the whole X-shaped structure would be the chromosome. If you put a little dashed line here, each half of the X-shaped chromosome would be a chromatid. Sometimes they're referred to as sister chromatids. The two chromatids are connected by a centromere, and these little black structures that you see on the edges of the centromere are the kinetochores. Okay, and the kinetochores are the structures that allow the spindle fibers to attach to each chromatid. Okay, now if we go down to the next picture, we have a picture of some chromosomes that are in a structure made of microtubules. Okay, you can see this little label over here, polar microtubule. All of these spindle fibers, or all of these microtubules together are making the spindle. Okay, so they're referred to as the spindle fibers. Okay, and the spindle is the structure that allows the chromosomes to move to the midline, and then once the the centromere breaks down, it allows the two chromatids to move to opposite sides of the cell. Okay, so this is going to be arranged so one end is close to the plasma membrane on one side of the cell, and the other end is close to the plasma membrane on the other side of the cell. Okay, and inside these kind of reddish areas that you see here that are at the tips of the spindle on each side, you have what is called in your book a centrosome, and it used to be called a centriole pair, so that's the term you may be more familiar with. And each pair is at a pole of the cell, and it arranges little fibers around it that stick to the plasma membrane. So this is what holds the spindle in place, and gives it an anchoring point so it can make the movements of the chromosomes that it needs to make. Okay? We also have a picture here of cytokinesis starting, and this indentation associated with cytokinesis is the cleavage furrow. And you can see the chromosomes are starting to be pulled away from each other. I should say the chromatids, once they're separated from each other, they call them separate chromosomes again, and they're being pulled away from each other here, so you can see that happening. Okay, so those are our structures. Let's go through the basics of mitosis. So our interphase cell, we can see here, there are two sets of centrosomes here. They're starting to move to opposite poles of the cell, and they're developing spindle fibers that are starting to move out around them. Okay, as they move further away from each other, the spindle fibers get longer, and you can see the genetic material is getting easier to see, more distinct. Okay, so it's converting from chromatin to chromosomes. As we go further through prophase, which is once the DNA is starting to condense, we say that we're in prophase. As we start going further, we see that the nuclear structure breaks down, the nucleolus is gone, the nuclear envelope starts to break down, the centrosomes are now at opposite poles of the cell, and the spindle fibers are getting really long. Okay, here we have almost no nuclear envelope left. Okay, and by here, the spindle is completely formed. The chromosomes are completely present. All of the chromatin has condensed into chromosomes, and we're at the end of prophase. Okay? Once we hit the end of prophase, the spindle fibers start pulling on the chromosomes and move them to the midline. Once they're all at the midline, we say that the cell is in metaphase. Okay, and you can see the beautiful spindle that you have here that's extending from the poles and surrounding those chromosomes. Okay, now at this point, the spindle fibers that are attached to the kinetochores start to put some tension on the chromosomes. And when that happens, the centromeres break down. And as the centromeres break down, the two chromatids start getting pulled away from each other. So they're starting to get pulled away a little bit here. They're a lot more pulled away, as you can see here. Okay? And they're moving towards the centrosomes at each pole. Okay, you can see the spindle fibers in between there. Then at the end of anaphase, we start to do pretty much the opposite of everything that we did in prophase. We bring our nuclear envelope back to surround our new chromosomes that we have here. We have our nucleolus start to come back. The spindle is still there, but it's going to start to degenerate. And we also see that the membrane is starting to pinch in so that we can separate these two new cells from each other. Okay, so this is telophase. As telophase continues, the nuclear structures in each cell become more obvious. The centrosomes start to lose the microtubule arrangements that they have around them, and you'll start to see a distinct line forming between the two cells. That line will get sharper and sharper, and eventually you'll get to the point where the membrane has completely sealed between the two cells and they're now separate entities. Okay, when the chromatin is reformed, the nucleolus is back and the cells are separated, we say that we've moved from telophase into a new interphase for each of these cells, and we're ready for them to grow and maybe go into mitosis again in the future. Okay, so if you look at this cell, This was the original cell. And then you look at these two daughter cells, as they're referred to here. You can see that each one is about half the size of the original parent cell. So it really hits home to you when you see this, why they need to go into a growth phase first, because they're just tiny. Okay, so they need to accumulate material so they can do the job they're meant to do in the body. Okay, so that wraps it up for this week. See you in lab.